Ayubovan to all of you, and good morning as well. And thank you all for coming. And I know that it's such a difficult time in Sri Lanka, and my heart goes to all of you there in Sri Lanka, because it's currently it's really politically and economically very unstable. And once you're born in Sri Lanka, your DNA is Sri Lankan. So we are all with you in Sri Lanka. And although we are not physically with you, we are mentally with you. So today I'll be talking to you about the work that's happening in my lab on the use of liquid biopsies in many cancer types. But because of the time today, I'll only be touching on head and neck cancers. This is my disclosure statement. The funding, we get national funding from the National Health and Medical Research Council, Australian Research Council, industry funding, Wyoming, Cancer Australia, and also US NIH. And these are some of the patents that have been licensed to companies. This is late Jake Simpson. About 4 years ago, at the age of 22, he had a severe toothache. He then went to see the dentist, and the tooth was extracted. But he continued to have pain. Then he was referred to ENT, and ENT consultants found a stage 4 cancer. Sadly, within 6 months of diagnosis, he passed away at the age of 23, leaving a very young family behind. That is how aggressive these head and neck cancer tumors are. And you may wonder why I picked head and neck cancers. Is because about a decade ago, my brother-in-law passed away with the same cancer, being diagnosed within 6 months of diagnosis at the age of 37. So I'm committed to developing early diagnostics to early detect head and neck cancers. I have 2 parts to my presentation today. So I'll go over this library diagnostics in terms of the biology and commercially available saliva devices, applications in my lab. And I'm not sure whether you've heard of exosomes. These are extracellular vesicles, and these are the connectors between different types of cells. So a cancer cell can dictate a healthy cell to become a cancerous cell, because the cargo in a cancer cell can be transported into a healthy cell. So it's like cancer to me is like an infection. In the second part, I'll talk about the liquid biopsies that's specially used in circulating tumor cells for applications in head and neck. I wouldn't be showing lung cancer and glioblastoma, which is the brain tumors. This is a research program. 2 parts: diagnostics, library diagnostic, blood-based diagnostics. And under this library diagnostic, we are investigating early diagnostics, predictive prognostics for head and neck, lung, glioblastoma. We are also discerning the oral microbiome, so the bugs in your mouth and its association with head and neck cancers. We are also developing saliva-based diagnostics to early detect and predict outcome in heart failure patients. Another program one of my PhD students is working on is developing a saliva-based test to early detect liver fibrosis. Liver fibrosis then leads to liver cancer, which is very common in Sri Lanka. And very recently, I think last year, we got a huge, massive grant from Australian Department of Defense to develop a denture so that you can monitor real-time stress markers. You can monitor fatigue in Australian soldiers. So this work is ongoing. In terms of blood diagnostics, we are investigating the circulating tumor cells. So these are the cells that are disseminated from primary and metastatic sites. And we are investigating their expression levels, as well as we are culturing them to do drug testing. As Preeti mentioned, I think the Air Force, they know about precision medicine. And when President Barack Obama came into power in 2015, his mission statement was to enable a new era of medicine through research, technology, and policies that empower patients, researchers, and providers to work coherently together to develop individualized care, which means the right drug for the right patient at the right time. That is precision health or precision medicine. I'm sure you all know who this is, Angelina Jolie. A couple of years back, she came out and she had mastectomy because her mom had, because she had BRCA1 mutation, and which predisposes a woman to developing breast cancer. So she used a preventative approach to avoid developing breast cancer, which is such a nasty cancer. Let's look at whole mouth saliva stored at room temperature under a cryo SCM. So you can see after 7 minutes, it's as saliva ages, it forms a mesh. Saliva proteome, just like the plasma proteome, consists of almost over 3,000 protein, and these include bacterial, fungal, viral, as well as human protein. Most of the major proteins are mucins, amylase, statins, instatins, IgG, soluble IgAs, human serum albumin. And just like the plasma proteome, most of the proteins that we are interested, predominantly cytokines, they're in the picogram per mil range, which means that we need sensitive detection technologies to early detect these biomarkers. Why saliva? I don't know you, I hate needles. The saliva collection is non-invasive, and with COVID-19, we saw how important it's to have field testing to stop the spread of COVID-19. And it's easy to transport, and anyone in a rural or remote community, you can collect saliva, and it's ideal for challenging clinical environments. Let's look at the saliva test in the market currently. Saliva hormone test, cortisol test, drugs of abuse, and this is one of the FDA approved saliva collection devices from DNA Genotek. HIV detection. This is the first FDA approved saliva-based test to detect HIV antibodies. And in Australia, we can buy it for about 50 Australian dollars over the counter. This is an Australian invention, which we are really proud of. It's a saliva glucose test. So, you know, if you know people with type 2 diabetes, or for that matter, type 1 diabetes, you have to keep pricking your fingers to know the glucose amount, either to administer your medication, which is metformin, or to administer insulin. And these Australian researchers have developed a saliva-based glucose sensor. And I think it was developed in the University of Newcastle, and the company is further developing it into a point-of-care device. 23andMe and Ancestry.com are where you can send your saliva sample and track your family tree, which is very common here in Australia because most people are migrants here. This is what I call 15 minutes of fame for saliva. FDA approved first at-home saliva collection test for COVID-19. And if you are in the US, you walk into Walgreens pharmacy and you can buy a at-home COVID-19 saliva test kit. We are also working in this space, and this is my postdoc, and he's looking into developing a saliva-based test for COVID-19. And we have 2 papers under review currently. Head and neck cancers, very awful cancer type to have. And it's the 7th most common cancer, as you can see. almost closer to 900,000 new cases, and half of them die within 5 years. Risk factors include smoking, alcohol consumption, HPV infection. One of the greatest problems the clinicians are faced with, they can't stratify the risk of these head and neck cancer patients. In other words, to identify who will develop recurrences from those who don't develop recurrences. So which means prognostic predictive biomarkers. This work is from one of my PhD students. As you can see from his name, he's a Sri Lankan student. What we did here is that we collected saliva samples from 268 patients, and we were interested in detecting HPV 16, which is the high risk common type. I know from Preeti's work, HPV 18 is the common type in Sri Lanka and in India. We collected tumor, we collected clinical information, we ran a qPCR, and I have a collaboration with a company called Agena Biosciences, where we use a mass array to confirm our findings. What we saw is that high-risk HPV types were detected in 72% of the oropharyngeal cancer patients. And as you can see, majority of the cases that were HPV positive are derived from the tonsillar and base of the tongue. The most common type, as I mentioned earlier, is 16, and tumor P16 was observed to be overexpressed in 89.4% of oropharyngeal cancer patients. So just let's look at tumor P16 is the current gold standard, and saliva HPV test is what we are advocating it to go into clinical practice. And if we look at the positivity, we get 99.48% positive, and negative is about 0.52 based on tumor P16 expression, which is quite good. And we were interested in cancer-specific survival. What you see here are the Kaplan-Meier curves. The blue color represent the positive for high-risk HPV. Red color presents negative for high-risk HPV, and you can see that if you have high-risk salivary, high-risk HPV, prognosis is better. P16 positivity, you look at it, it's also better, but if you combine salivary high-risk HPV with tumor, it's much better, which is the blue color one, the top one. I'll talk through our world first discovery, which next week we have been shortlisted by the hospital for an award. We'll wait and see whether we get it or not. And this work is done with my collaborator, Dr. Saj Vasani is an ENT consultant, and my I think we've been collaborating for over 10 years, Liz Kenny, who's a senior radiation oncologist. And this work was done at the University of Queensland Dental School, where we had over 600 dental patients. And the reason why we picked UQ Dental School is that because most people, low socioeconomic people, come there because they get free dental treatment. And we collected saliva at baseline, and we followed them every 6 months. Out of the 600 patients, we had 11 of them to have HPV positivity in saliva at one time point, 3 of them to have persistent HPV. And we know that persistent HPV is a precursor for developing HPV-driven oropharyngeal cancers. So for me, when I looked at that data, even long after the trial has ended, I felt like, even though this is a research trial, these people are at risk of developing this cancer. And I'm a strong believer of karma. And I thought, I talked to the clinicians, and it took us, believe it or not, 8 months to go back to ethics and get ethical permission under the what is called clinical duty of care. And we brought all three. Lo and behold, this person is asymptomatic 60-year-old male. And as you can see, the HPV copy numbers increase. And this point is the point when he was referred to ENT. He had a normal MRI done. MRI was negative, and he had an endoscope done. The two tonsils, the left one was quite enlarged. And Saj offered him a tonsillectomy. And he said he decided to go with a tonsillectomy because he's a teacher who's read about oropharyngeal cancers, and he had all the risk factors. And once Saj operated on him, and we were able to detect a 2mm occult cancer at the back of his throat. And what you see is that after 2 weeks, I went myself to collect the saliva sample from this person, and the HPV copy number was 0, which means that we have closed the loop. We have found the cause of the cancer, and if we use HPV, we can monitor, and we can use HPV to early detect recurrences, which one of my clinician PhD students is currently investigating. This is the work I did with a company based in US called Viome, where we investigated the microbial gene expression and linking it to oral cancer. And if you are interested, you can read this paper, and it's open access, so you can access it. If you look at the control group in blue versus oral cancer group, they do separate, but they did a lot of artificial intelligence and machine learning algorithms to come with a signature of 700 gene signature to predict outcome. This is investigating salivary microRNA. So because we are interested in early detecting cancers, we are interested in epigenetic regulation. As such, we are investigating DNA methylation. That's a work I did with Preeti, and microRNA. What you see here is in the control group, because it's qPCR normalized to the control group, you can see different microRNAs elevated in saliva. These are the area under the curve. If the area under the curve is 1, that means 100% sensitivity, 100% specificity, which you will never find in real life. And this work I did with when I was doing a sabbatical at Hopkins. And that's the time they had TCGA data. And I worked with the bioinformatician to download TCGA data for head and neck cancer to see the markers that we identify, what's their expression in tumor. Blue is normal, green is tumor. So it's very important that you pay attention to this figure, because on this left side is the data generated in an Australian population. On to the right is the data generated in a US cohort. And you can see, let's look at MIR9 is elevated in the tumor compared to normal 191, it's also the same. Then we were interested in investigating microRNA to see if we have a cohort of healthy people who are persistently positive for HPV, can we discriminate that from HPV positive oropharyngeal cancer patients? Because what we want to develop is a screening tool. So a screening tool is used within a high-risk group. So the high-risk group will be persistent HPV, and we can predict the likelihood of developing cancer. And that's exactly what we did. We did bulk sequencing, identified microRNA, and Chamindie validated all these microRNA, and out of the microRNA, 3 of them, as you can see, with a very good area under the curve, sensitivity and specificity. And this paper currently is under review, And we also have filed a patent with Chamindie. Exosomes, as I mentioned, are extracellular vesicles, and we are using exosomes as biomarkers. This is just to demonstrate that if we collect different types of saliva, this is drool saliva versus acid stimulated versus oral rinse. So oral rinse saliva is like a swish and gargle. When you get a sore throat, you get salt water and do a swish and gargle, and that's the oral rinse sample. Think of acid stimulated saliva as sucking onto a lemon lolly, and drool is resting saliva. And we investigated the protein abundance, and you can see that the drool has more protein compared to the other two. And these are the markers that characterize exosomes, and you can see that these exosome markers are present. And these are, the exosomes are like cup-like, and this is what you can detect. Proteomic signature is also different between the control and the patient group. And this work was done using mass spec. Now shift gears to liquid biopsies. So liquid biopsies is the analysis of biomarkers in body fluids, in our case, saliva and blood, and we are slowly moving into urine, and we're investigating circulating tumor cells, exosomes, DNA, microRNA, everything else. So you may ask, why liquid biopsies? Tissue biopsies are invasive, and some locations are difficult to access, such as in non-small cell lung cancer patients and brain tumors. It's very difficult to access a tumor tissue. Single biopsy can miss relevant tumor clones due to intra-patient's tumor heterogeneity. Sequential tissue biopsies is not possible with surgery, because once you take the tumor tissue out through surgery, how do you monitor the response to treatment in a cancer patient? And that's through the use of liquid biopsies. Liquid biopsies are CTC, ctDNA, methylation, circulating microRNAs, and EVs. CTCs are the cells that are shed from primary and secondary or metastatic sites, and they are the surrogate markers for overall survival and progression-free survival. If for breast, prostate, and colorectal, and this work has been published in New England Journal of Medicine about 10 years ago. Among the cells that have detached, only 0.01% can form METs. And CTCs are very rare. It's like 1 to 10 CTCs per ml of whole blood. This work is done in collaboration with one of my long-lasting collaborators from University Technology Sydney, Professor Majid Wakani, and we use his chip, and we isolate, we collect blood, do red blood lysis, and we isolate cells based on inertial microfluidics. I'll play this video for you now, and you can see the cells that move up are the so-called CTC. And the way we characterize CTCs got to have a DAPI, the nuclear staining, cytokeratin positive, and CD45, which is a leukocyte marker, should be negative. And we are using that for drug testing. This work just got last week accepted in cellular oncology. This work is done by Z, who now came and helped me. And this is a longitudinal trial. We will close the trial in June, where we have collected 119 patients prior to treatment, after 13 weeks when they come for PET-CT, and we correlate PET-CT data to the baseline data 6 months to 24 months. And what are our clinical endpoints? Complete response within 24 months and incomplete response. So if you look at the CTC number, complete responders, 108 have this number of CTCs, and incomplete responders, N equals 11, have CTC. So CTCs, if you don't respond to treatment, 90% of the time we can detect CTCs, which means CTC can be used as a rule-out test in clinical management. We are also deriving CTC in culture, and we are interested in doing drug testing because, as you may know and appreciate, most of the time, if the drug doesn't work, the comorbidities associated with drug would kill the cancer patient faster than the disease could kill. And we want to do a personalized approach where we will test the drugs in CTCs prior to giving it to the patient. So we have developed 12 CTC cell lines, which have been now, 5 of them have been sequenced. We sequence their primary tumor, we sequence their CTC cell line, and that's what you see, data for 2. The common mutations between primary tumor sample and cultured CTC. So these are COSMIC and CIVICA2 databases. And we have one gene that's a mutated gene that is common for these 2 patients, but you can see different mutations. So in other words, to tell you simply, we confirm that the CTCs that we culture are derived from either primary tumor tissue of the same patient, which is a big milestone. We are now characterizing these CTC cell lines. So in summary, I think I have convinced you that saliva would be, I believe in the next 5 years, it will be a tool in clinical management. And in fact, currently we are providing salivary HPV testing to the clinician for clinical decision making, especially when they have unknown primaries and they don't know where to look for the primary. If the saliva is HPV positive, they straight away go into the tonsil area and they can detect the cancer. See how simple it is and how it can revolutionize the clinical management. Microbiome changes can predict oral cancer, and we are continuing to working with the company Biome in developing a next generation of diagnostics. MicroRNA levels or changes in saliva can be used to early detect head and neck, and I've shown you that CTCs can be used as a prognostic biomarker in head and neck cancers. With that, I would like to acknowledge my team. So I can tell you it's quite a diverse team. Tilani, Basnayaka, Chamira, Ekanayaka, and Nuan are my Sri Lanka. So these are all my students, but now Nuan is working as a clinical trial coordinator, and Chamira is just working as a research assistant until he gets his PhD results. With that, all collaborators and funding bodies, I thank you, and I thank you for listening. Happy to take questions.
Thank you very much, Professor Punyadeera. It's such a interesting seminar. I think I'm audible, everybody, right? I'm in the field, so still a bit struggling with the connectivity. Yes, thank you very much, ma'am, for the presentation. Now the floor is open for questions. We can, I think, Professor Punyadeera can take few questions. The floor is open. Actually, while people are getting settled, Professor, I have a question. We normally consider saliva as a PCR inhibitor when we do sampling blood or sampling tissues. We try to not to get contaminated, saliva contaminated, contaminating our tissues. I'm talking about blood, like animal blood tissues and samples and so on. Partly because of the DNAs and RNAs in it. How true that is? So when you deal with the saliva, like say HPV and such DNA, do you have to have specific conditions to not to worry about these RNAs and DNAs?
So it's a very good question, and thank you for that. Yes, so there are stabilizers, just like for any blood or anything. We use Kizol or a triazole, which inhibit all RNases. So for RNA work, we use RNA later, which inhibits RNases. So we don't have any issue. We are now trying to see whether we can post these saliva samples from rural Australian outback into our lab, and it's another project that we have recently started, and we believe it can, because Viome is doing that. It's just a RNA DNA shield or a protect, which protect the RNA and DNA by stabilizing them as well as deactivating the enzymes.
I see. Great. And another related question, just a small one. So can you go up to next-gen sequencing, whole genome sequencing level using saliva, or do you still have to rely on conventional tissues on that?
So the work that Viome did, and that's on the microbial part, and that's next-gen sequencing, and it's microbial transcriptomics, and we have done bulk sequencing as well on saliva. Because what we do is that we take the saliva, isolate RNA, and then we prepare the libraries and do sequencing.
I see. Okay, thank you so much. Yes, the floor is open for questions.
Chamindie, congratulations, hearty congratulations all on the wonderful work that you presented. Just an unrelated question. I'm just wondering whether, now COVID-19 can be detected through saliva. It's basically antibodies, right? Chamindie?
Yeah, I'm listening.
Yeah. So likewise now, when the animals are being screened for COVID-19, has anybody used mouthwashes or saliva or the like? Would you know?
I wouldn't, as you know, I'm only with the humans. I've got no idea about, I haven't read about animals. But the thing is that COVID-19, it's like the SARS-CoV and things like that. If it's there in a cat or a mouse, or you should be able to detect it. But I haven't read on that. I don't know the answer.
Okay, all right. And how effective is saliva diagnostics on COVID-19 testing?
Saliva, I think you must have read the Nature Medicine and Lancet papers that came earlier on. They showed that when a patient is COVID-19 clear using a nasal swab, yet saliva was positive. So literally people were released into the community thinking that they are COVID-19 negative. Meanwhile, the saliva was still positive.
Oh.
So it's highly sensitive.
Right. Okay. So for ELISAs, you have to use sort of enhancers to detect the, say like biotin, iodine or something like that, or in a direct ELISA. I'm sorry, I haven't read much. I have to confess.
It's just a PCR.
Oh, only a PCR. Okay.
Yeah.
Right. Okay.
Because
so what about the antibody stuff?
Antibodies are done with ELISAs. I haven't done antibody work. We do PCRs.
Okay. Right.
So, yeah.
Yeah. Right. Thank you.
Thank you very much, Prof. Chamindie, for that interesting presentation. I have a small question.
So you mentioned that you have found several CTCs in relation to head and neck cancers.
Mm-hmm.
So do they have any genetic similarities to the CTCs that can be seen in other cancer types as well?
So it's a great question. So we do CTC work for glioblastoma, that's brain tumor, lung cancer, right? A CTC by definition, if you ask me, is a cell, it's an epithelial cell, right? These cancers are epithelial. Cytokeratin positive, DAPI positive, CD45 negative. However, a lung cancer would have a EGFR 19 or 21 deletion compared to overexpression of EGFR in head and neck versus GFAP. overexpression in brain tumors. So we can actually, if we use those markers, we can say whether it is a head and neck, glioblastoma, or lung cancer.
Okay. And can I also ask a very practical question, like when it comes to practice, do we have any hindrance to collect samples from patients, like let's say people who are chewing beetles or taking any type of drugs, or do we have any kind of issues related to these people?
The question is, sorry, you broke up. The question is, are there effects of drug on saliva collection, or
effects of drugs or chewing beetles, people chewing beetles? When you are collecting samples, do we have any?
Yeah, okay. What we do is that when, let's say a person is chewing betel nut, right? The whole mouth is red. What we get them to rinse the mouth first, because we don't want all that red color in our saliva sample. We'll make sure that they wash, but we ask them not to brush, because if you brush and if you have leaky gums, that means that blood molecules can seep into saliva. So there is already contamination.
Okay. Okay.
So luckily here we don't have the betel nut issue, so it's all good.
That's why I asked when you have to practice something in Sri Lanka, the environment is.
It shouldn't be a problem. We just get them to rinse their mouth, or ask them not to eat betel nut for 1 hour. I'm sure they can stop.
Okay, thank you.
Ma'am, I have to speak one. So saliva, so such a broad spectrum kind of a tissue for somebody like you to develop tools. Would other body tissue fluids like mucus from phlegm, from genitalia, breast milk, are those have the same potential in your opinion, or saliva has something special that you had happened to tap on?
So no, it depends. Let me take a step back. If, for instance, a prostate cancer, if you are developing prostate cancer biomarkers, you need to use a body fluids that's adjacent to that cancer type, which is urine, right? So yes, urine biomarkers, PSA in urine, right? Or blood, whichever way. And people are also using for eye diseases tear fluid.
I see.
But I think tear fluid is very invasive, because you go with a little pipette type thing, and you have to take saliva is like anyone can drool, anyone can give a saliva sample, because the urine, you really need to have a bathroom for people to give you a urine sample, but saliva can be collected in the field.
I see. And sweat, would sweat?
Yeah, so I told you in the beginning that we got a grant to develop a saliva denture, and we are also developing a wearable sensor based on sweat.
Sweat. I see. Got it. Thanks. Yeah. Thank you so much.
Madam.
Hi, hello.
Hi, madam. Praneeth from Professor Preeti's lab.
Oh, hi, Praneeth. You know that you shouldn't call me madam. You know this, right?
Yeah.
Okay.
Oh dear.
Hope you are doing good, madam.
Yeah, and how are you doing?
We are doing good.
Yeah, go ahead, Praneeth.
When we are into salivary markers, how the oral microbiome can affect the results of salivary markers?
It's a coexistence, right? The primers are specific. If your primers are specific for the human microRNA or methylation, it should not interfere with the microbiome thing, because it's selectively amplifying. Is that what you're asking for?
Madam, actually, how this symbiotic relationship or the effect of microbiome can does it can influence the RNA expression of these particular cell niches and?
Yes. And that's why we are trying to investigate which are the bugs that cause cancer. It's like a chicken or egg, right? Does the cancer come first, or the because of the cancer the oral microbiome profiles change, or it's the other way around? No one knows thus far. You want to do a PhD on that?
Good idea, Venus.
We'll talk offline.
Thank you so much.
Yes, I think, thank you so much, Professor. It's very exciting, very proud personally to see somebody from Gaul, southern sector, somebody from Sri Lanka, somebody from Asia, leading the way in opening windows for humanity to prosper, to have a better quality of life, better future for the entire humanity. And with that, I will invite Dr. Sachini Amarasekara. We do this like Dr. Sachini and Dr. Maheshi Mapalagamage, three of us normally run around and with the help of the FOSS lab, in this case, Professor Udagama's lab. So Sachini, over to you to formally thank our speaker for this session. Over to you, Sachini.
Thank you, sir. So on behalf of Ministry of Nerds, let me thank Professor Chamindie Punyadeera from the Griffith Institute of Drug Discovery and Menzies Health Institute, Queensland, Australia, for talking to us about liquid biopsy for precision health in head and neck cancer, which I think is very timely and important because non-invasive procedures for diagnostics, therapy and monitoring is now preferred by many patients and practiced by scientists and clinicians in most part of the world. So I think this will be an eye-opener for our scientists and clinicians as well. And most importantly, you have shown how the work we do on the workbench can be brought into a clinical practice. And thank you very much for it. We are really grateful to you for joining with us today and inspiring all of us. Thank you very much, Professor Preeti Udagama, Head Cadre Chair of our department and the Principal Investigator of the Combinatorial Research Lab of the Department of Zoology and Environmental Sciences, and her team for hosting today's seminar. Finally, thank you very much, everyone, for joining with us. Stay safe and have a pleasant day.
Thank you so much, and stay safe. Take care.
Thank you so much.
Bye.
Bye-bye.
Bye, Chamindie. Thank you so much.
Bye.
Bye. Bye.
Bye, Casey.
Bye.